Hey folks, welcome to your last set of Chemistry 2 video notes. Today we'll be covering the last of our 4 classes of biomolecules, lipids. By the end of this lesson, you should be able to recognize the basic structure and function of the 3 main types of lipids and, based on that structure, determine whether a triglyceride would exist as a solid or a liquid at room temperature. So, we've learned about proteins, nucleic acids, and carbohydrates, but there's one more class of biomolecules that we still need to talk about: lipids. Lipids are all the biomolecules that are not soluble in water. In other words, they are soluble in nonpolar substances. They're much smaller than complex carbohydrates or proteins, so structurally they're a little easier to picture. There are 3 main types of lipids: triglycerides, also known as fats, which are used to store energy; phospholipids and glycolipids, which are found in cell membranes; and steroids, which are molecules that are based on a particular 4-ring structure and include important substances like cholesterol, hormones, and bile acids. Let's talk about triglycerides. Triglycerides are the most abundant of our 3 types of lipids. They're used for long-term energy. To give you an idea of what I mean by long-term energy, think back on our last set of notes when we talked about glycogen. We learned that glycogen is stored in the muscle tissue and can provide energy to the human body for about a day. Triglycerides are stored within cells, and depending on how much of them is stored in the body, they could provide energy to the body for months. Triglycerides are derived from fatty acids, which are long, unbranched hydrocarbon chains with a carboxylic acid group on one end. Look at the picture of glycerol here. Glycerol is an alcohol made of 3 carbons, each with an OH group attached to it. Triglycerides are just esters of that glycerol molecule. Simply, those long-chain fatty acids will bond their carboxylic acid carbons with the oxygen in the glycerol's OH. When all 3 of those oxygens in glycerol have fatty acid bonded to them, we get a triglyceride, like the one pictured here. The R in this picture just stands for the rest of the fatty acid chain. We could have any 3 of our multiple fatty acids bonded here, but this part of the triglyceride will always look the same. As you just saw, triglycerides are made up of a glycerol molecule attached to 3 fatty acids. This could be a simple triglyceride where all 3 fatty acids are the same, or a mixed triglyceride, where there's 2 or 3 different types of fatty acids used. Either way, the shape of those fatty acids makes a big difference in the properties of the triglyceride. If the fatty acids in the triglyceride are saturated, that means they have all single bonds and they are linear. This linear shape of the hydrocarbon chains provides more surface area and a less clunky shape for the triglycerides to be attracted to each other via intermolecular forces. In other words, if the fatty acids in our triglyceride are saturated, the triglyceride is going to have a higher melting point and will be a solid at room temperature. We call saturated triglycerides fats. If the fatty acids have double bonds that are in the cis orientation, the shape of the fatty acids are no longer linear. This nonlinear nature of the fatty acids creates a much clunkier molecule, one that isn't easily as affected by intermolecular forces. In other words, the more cis double bonds the fatty acids in a triglyceride have, the more likely that triglyceride will be a liquid at room temperature. We call these unsaturated triglycerides oils. Here are 2 examples of triglycerides. Figure A is made of saturated fatty acids, while figure B is made of unsaturated fatty acids. You can imagine that the saturated triglyceride in figure A will be able to line up with other molecules like it and have higher intermolecular forces of attraction between them than the unsaturated triglyceride in figure B. Generally, saturated triglycerides are solid fats and are less healthy than unsaturated triglycerides, which are usually liquid oils. This is because the saturated triglycerides tend to have an easier time forming solid fatty deposits in the body, whereas the unsaturated triglycerides are less likely to form deposits, as they aren't as highly attracted to each other. The only exception to this rule is trans-unsaturated triglycerides. These are triglycerides in which the fatty acids have double bonds, but those double bonds are in the trans position. This still gives us a fairly linear molecule and will act like a saturated triglyceride, or a fat instead of an oil. Both oils and fats are stored within cells until they're needed later for energy. Our second type of lipids are phospholipids and glycolipids. They're used to form cell membranes. There are many types of phospho and glycolipids, but the most common ones are esters of glycerol, just like the triglycerides you just saw. Phospholipids are found in animal cells and contain a phosphate group and usually an amine group. Phospholipids look like this, where you have two long-chain nonpolar hydrocarbons in the first two esters, but the third group is a phosphate group with an amine attached where that X is. This phosphate-amine group is highly polar. Glycolipids are very similar, but instead of a phosphate group, they have a galactose sugar molecule in its place. These are found in plant cells. In both cases, we have one end of the molecule that's very polar, where the phosphate or galactose is, and the other end is very nonpolar. This causes them to form in a double layer when forming cell membranes. In this double layer, the nonpolar ends are tucked away on the inside, so they don't have to interact with the water outside the cell or the aqueous cytoplasm inside the cell. That pushes the polar ends of these molecules to the outside and makes it look like this picture here. Well, today's your lucky day. You get a bonus group of lipids called waxes. These are esters of fatty acids and any other alcohol than glycerol. They're often found on the outside of plants and animals, as in protective coating on leaves, fruits, skin, fur, or feathers, or as in beeswax. The condensed structural formula of beeswax is shown here. Our last group of lipids are steroids. All steroids have the same basic 4-ring structure pictured here. There are 3 6-membered rings and 1 5-membered ring. There are a number of different types of steroids, but a really important one are the sterols. Sterols have an OH group on a specific carbon in that first 6-membered carbon ring. The most common sterol is cholesterol. Cholesterol's purpose is to transport fatty acids through the body. Another type of steroid are the steroids in bile acid. Bile acid is made in the liver, passed through the gallbladder, and lands in our small intestine, where it helps digest our food. 2 of those steroids are pictured here. Lastly, most animal hormones are steroids as well. Hormones are responsible for a lot of different physiological processes. Though they exist in very low concentration in our bodies, they produce some highly pronounced physiological effects. They can affect reproductive systems, emotions, body development, and more. 4 hormones are pictured here. Take a moment to analyze the structures of these 4 hormones and the 2 bile acid steroids. Notice they all have that 4-ring system within their structure. The major differences are what's attached to the carbons in those 4 rings. So, to sum it all up, lipids are all of the biomolecules that are not soluble in water. These include triglycerides, phospho and glycolipids, waxes, and steroids. Triglycerides are esters of fatty acids and glycerol. If they're saturated, they have no double bonds and exist as a solid. We call them fats. If they're unsaturated, they have double bonds, and if those double bonds are cis bonds, they exist as a liquid, and we call them oils. Phospho and glycolipids are found in cell membranes of animals and plants, respectively. They're just like triglycerides, but one of those fatty acids is replaced with a phosphate and amine group for phospholipids, or a galactose and amine group for glycolipids. Waxes are esters of fatty acids and any other alcohol other than glycerol. They're found externally on plants and animals. And lastly, steroids all have the same basic 4-ring structure, with other groups attached to it. Among the steroids are the sterols, like cholesterol, that have an OH group attached to a specific carbon on the first carbon ring, as well as bile acids and hormones. Well, that's all for today, folks. I hope this video helped shed some light on our 4th class of biomolecules. If you have any questions, please let me know.